Excited to be here today and provide an update on Miromatrix. We've had a tremendous last 12 months, so it's going to be exciting to just talk about some of the exciting updates that we've had over this time. One of the first ones is starting with we had our successful IPO last June, so that now means that I need to provide this slide each and every time. With that, though, to introduce you to Miromatrix, for those who don't know the story, whether in person or virtual, Miromatrix is dedicated to eliminating the organ transplant waiting list. We all know the numbers that are out there, and these are just US numbers. Over 115,000 patients today are waiting for an organ. The number that I gravitate towards a lot on this is actually 70,000 patients, and that is 70,000 patients this year will go to their physician, be pre-screened for an organ, be okay with immunosuppression, but not have the ability to be able to receive that organ this year. That's really what we're looking to change. We are a tissue engineering company with our technology to be able to bioengineer transplantable organs. And our goal is not to just stop at eliminate the organ transplant waiting list, because the true need that's out there far exceeds that as well. Throughout our history, and I'll get into this a little bit in terms of our updates and our technology, we've attracted key strategic investors, including DaVita, Baxter, CareDx, as well as great scientific collaborations with Mayo Clinic, Mount Sinai, and Texas Heart. This is just a quick video real quick, just introducing our technology.
Our patented perfusion decellularization technology starts with a functional organ from a pig that would normally be discarded. Next, we remove all the organ cells by first cannulating the organ and then flushing it with a simple solution that only removes the cells, resulting in a pristine decellularized organ matrix that can be stored for over 12 months. Next, the decellularized organ is placed into a bioreactor, and we initiate our patented recellularization process. During this step, human cells are introduced to the organ, and over the next 14 to 28 days, the organs are continuously monitored and perfused with the appropriate nutrients and oxygen to achieve the desired key growth parameters, resulting in a bioengineered organ.
So hopefully that gives you a good overview of the technology, but if we break it down just a little bit more, what you saw in there is the process is two-part. First, we start with an existing organ. In this case, we take an organ from a pig that would have been naturally discarded or used for other things, and then we use that process, perfusion decellularization, to remove all the cellular material. Early on in our development, we wanted to make sure that that material, or that scaffold that we start with, was not immunogenic, so we actually commercialized that product. We took whole porcine livers, perfusion decellularized those, created a surgical mesh, got that through the FDA, manufactured that. That's been implanted in thousands of patients. So we've got a great safety profile with our starting base material. The next start of the process that you saw there was the recellularization. That's that ability then to add cells back in and bring that function back, but in this case, add human cells. So this allows us that platform to bioengineered organs that don't have any animal cells in them. So we do not believe that they're considered a xenotransplant. We've seen some of the recent news and the excitement around some of that as well to address this from a different angle through genetic modification of the pig to be able to bring those forward. One of the challenges with that is just the unknown and long-term immunosuppression complications that exist on that. We believe we bypass that or avoid that with our approach. Highly scalable, as you saw in the video, and you can see in the picture there, our manufacturing facility, where each one of these become individual closed systems to be able to manufacture. And then given that everything's the same on these organs in terms of plumbing, there's not a need to recreate the surgical procedure associated with them. Breaking that down a little bit more, because I get this question a lot, how long does it take to recellularize an organ? It takes about 2 to 4 weeks. Once it's decellularized, that scaffold's incredibly stable. We place that into a bioreactor. We then introduce the human cells, and over the course of 2 to 4 weeks, that's continually perfused. We add cells, nutrients, oxygen to bring that to maturation. The other question is, what type of cell source do we start with? And we look at this in 2 phases. The first phase, we actually source organs, primary cells, from organs that were not placed for transplant. What that allows us to do is start with our first product, and we're really focusing on those 70,000 patients today who want an organ. How can we get that to them as fast as possible? From a regulatory standpoint, we start with a matrix that the agency's already seen, as well as we start with a cell source that the agency's already seen in terms of primary non-modified human cells. Bring that first product all the way through, be able to save lives, and then focus on the second generation, where the long-term ability would be able to recellularize these organs with stem cells, patient-specific cells, bypassing that long-term immunosuppression need. As I mentioned, we just completed our in-house manufacturing capabilities. These are some pictures of that facility. What this really gives us is complete control over our manufacturing, really from the start to the finish of the process. The facility's been designed to be able to support us all the way up through manufacturing of our clinical product to be able to support our clinical studies. If we look at this, and I'll get through this in a second, what is our pipeline? Our focus is really to start with is on liver and kidney. Our first product is actually the liver, where we take that outside of the body and use that in a liver assist a product, followed by a fully transplantable liver and a fully transplantable kidney. We've made tremendous progress, especially on the MIRO liver ELAP. We submitted our pre-IND late last year. We got those responses back, and based on that response, we gave the guidance that we're planning to submit our IND later this year. So it was very favorable in that sense, and we're excited to move to that next level. The guidance that we've given on fully transplantable liver and kidney is we're targeting pre-IND submission early next year on both of those products. So quick overview. If we look at MIRO Kidney, just from a market opportunity standpoint, or just framing this up a little bit more on that, today, and these are just US numbers, we have 500,000 patients who are on dialysis today. 100,000 new patients are added annually. I think the thing that's surprising about this is that when you do look at dialysis, the 5-year mortality rate associated with it is 50%. We've talked a lot about cell therapy here, gene therapy. In some of these diseases, have mortality rates very similar to some of those other rare diseases or orphan diseases as well. So this is something that we're really looking to be able to address. And overall, there's a huge war on kidney disease. We see that with the executive order on advancing American kidney health that targets the development of an implantable kidney, as well as CMS's kidney choice model, which really provides financial incentives for people to propose and recommend transplant, which is great. The challenge is there's not enough organs to be able to transplant. So everyone knows that transplant is really that gold standard, but there's just not enough available. That's really what we're looking to solve here, as well as we're a winner of the Kidney X Redesigned Dialysis Award. So if we look at this a little bit, not to go too far into the data, but if we look at the recellularization, how are we able to introduce those cells back in there? We talked about this before, but that's the ability to get the vascular cells to localize to the vasculature. Glomerular cells, that's the filtration of the kidney, be able to demonstrate that we can get recellularization of that compartment, as well as a nephron. But then we can take that a step further, and in this first one, we recellularized a porcine kidney graft with human endothelial cells, placed that back inside of a kidney, and then did an angiogram 7 days later, and you can see that we were able to maintain that patency. This really solved the vascular challenge that was in tissue engineering, which really inhibited and limited the ability to create advanced tissues that then could support that higher cellular functionality. As well as we can take that fully recellularized kidney. In some of our preclinical studies here, you're seeing the clamps come off, and you're going to see that initial reperfusion of the graft there as well. We get great perfusion, but we can also start to look at that effluent, or early urine production that starts to come out of the organ in that, and the effluent's on track with what we see in a transplanted gold standard kidney today in that preclinical model. Today we focus, if you look at the histology, we're about 30% recellularized. What we're targeting is that 60 to 70% recellularization rate prior to going into human clinical studies, and we're making great progress on that. Our other lead program as well is the MIRO liver. And with liver, similar to kidney, huge need that exists out there. With liver, though, we have 40,000 patients who die each year of end-stage liver failure because there's no alternative therapies. There's no drugs, there's no devices, there's no biologics for these patients. It's either you're fortunate to get a liver transplant or you're not. And that's really what we're looking to solve here as well. But there's tremendous expansion opportunities as we really look at some of liver diseases that continue to evolve and come forward. On the liver, similar to what we saw on the kidney, we're able to recellularize this. So this is an image of, we've revascularized this. This is going back into a pig transplant here. A heterotopic transplant in this sense. You can see that initial perfusion or filling of the graft. It's still going to be white up on the top of that because we don't have that outflow clamp taken off. Once we remove that outflow clamp, now we have complete perfusion. So at this point, this has now been integrated back into the host circulatory system. So again, we solved that. We published that in Nature Biomedical Engineering. The other thing that we can do is now take liver-specific cells and we can recellularize that graft, and we can demonstrate in the bioreactor, those chambers that you saw in that first video, the ability now to produce albumin, to produce urea, but more importantly, we can start to challenge it with functional challenge assays associated with liver function, such as ammonia, where we can dose in high doses of ammonia, and we see beautiful functional clearance of that. That allowed us to take it a step further into a large animal preclinical model where we created an acute liver failure model inside of a pig model, where we essentially ligated off and took out the native liver and then heterotopically transplanted the bioengineered liver in this case, and then looked at it over that time. What happens when you take the native liver offline is you go into acute liver failure, and you get really high levels of ammonia that accumulate inside the blood, similar to what you see in acute liver failure. And eventually you get hemorrhage associated with that, or multiple organ failure. And then what we saw inside our model is when we introduced our liver grafts, in this case they're about a third of the size of the native liver, we saw that ammonia come up, level off, in some cases coming back down. So that function that we saw outside or inside the bioreactors, we're able to now replicate inside of our preclinical model in this case. And since then, we've increased the graft size, we've increased the hepatic cell mass in that as well to further bring down those ammonia levels. This really sets us up for our first study. This is what we filed our pre-IND on and planning to file our IND later this year, is a MIRO liver ELAP. And for that, with acute liver failure, there's no alternatives for this as well. If a patient presents with acute liver failure, unfortunately, 30% of those patients will die. 25% of those patients will receive a liver, and then 45% will naturally, spontaneously recover. So tremendous need in this is from a therapeutic solution as well to be able to come forward. And we see the perfect opportunity here to take our bioengineered graft, now place that external to the patient, because the liver is the one organ that can regenerate itself. And it's believed during acute liver failure, what happens is you really just need to offload the native liver to allow that time to be able to recover. So this would be an initial bridge to transplant type study. And what we want to do, be able to demonstrate in this is safety and tolerability, because that is a phase 1 clinical study. But at the same time, we're still going to be able to draw as secondary endpoints blood samples and look at that ammonia clearance and other things to be able to start to look at that effectiveness associated with our liver graft. And then utilizing that data to continue that product line, but also leverage that for our fully transplantable liver and kidney programs as well. So we've always had a very focused regulatory strategy to de-risk and accelerate towards human clinical data, and we've always had a patient focus to really get this to who truly needs it, and that's the patients. And that started with the approved matrix, as I described in the beginning, to the confidence in the decellularization and the ability to be able to create the perfect scaffold to bioengineer on, the ability to then be able to source the appropriate cells, then to bring back that function that you saw as well. That sets us up for our first clinical study that we're planning on the MIRO liver ELAP, and then that'll be followed up then by human studies that we're targeting for the MIRO kidney and the MIRO liver. With this, you always need a great team. Excited as we've been able to continue to grow this team as well. Senior management, led by myself. Jim Douglas is our CFO. Mason, regulatory and clinical affairs. John Barry, R&D, Logi, quality. People and culture, and VP of finance. So it really does take that team to be able to continue to move us forward. Phenomenal board of directors, as well as key advisors from great medical and scientific locations across the world. So in summary, really excited that we're really targeting what we feel is one of the largest unmet medical needs, which is transplant. We just haven't seen a lot of innovation in that space, and excited to be able to bring forward a technology and approach to be able to truly start to be able to address this. Incredibly disruptive technology. We have world-class in-house manufacturing to be able to do this ourselves. Very fortunate to have the strategic investors that we've had over the time, DaVita, Baxter, and CareDx. But I leave you with, we're really looking to change the world of transplant and the patient experience. We see a future that a patient isn't waiting by the phone for that call and then has to come in within the next 12 hours. Really see a future where patients can schedule their transplant well in advance and be able to come in and have access to that life-saving technology. So I appreciate the time today, and look forward to talking to anyone who has questions later. Thank you.